Controlling gene expression. In general, gene regulation means that the level of gene expression can vary under different conditions. Depending on what the cell is trying to do, it may need more or less of a particular protein, and those proteins are encoded for in the genes. And so if we regulate those genes, we get the theoretical amount, the right amount of protein. So again, we don't want all of our proteins expressed, all of our genes expressed, and all the proteins created all at the same time. Sometimes we want them not at all, and sometimes we want lots of them. So it depends on the conditions themselves, and therefore we want to have a way of regulating the expression of those genes, the production of that protein. So we will focus on prokaryotic transcriptional regulation. This is the most efficient place to do the regulation. And in order for this gene to be read, for the bacteria to take this gene and turn it into protein, there is a promoter region before it, and that essentially has to be accessible by RNA polymerase. It has to be able to bind there so they can begin the transcription. The regulatory proteins are things that will either help or hinder this from happening. The ones that help it are called activators, and they expose the promoter. They make it easier for RNA polymerase to land and therefore to make our mRNA. They turn transcription on. And then there are other proteins that act as repressors, and they stop RNA polymerase from binding there, and therefore the gene will not get transcribed. Genes that are constitutively on constantly on are called constitutive. These are housekeeping genes, so genes that need to be read all the time, proteins that's constantly being used, they have to be on all the time. And so transcription factors, the pieces that go about going to transcribe this, are going to be needed all the time. Regulated genes are either repressible, and so they can be turned off, so sort of they're normally read, but they can be turned off, or they're normally off and they can be turned on, so they are inducible. Negative control is when they are normally on and they get turned off. And so we have these genes that are normally being read, but under certain conditions they can be turned off. So since they're being turned off, this is called negative control. And one of the ways to do that is if, again, this is the segment of gene, there's going to be a promoter region. If this particular protein binds there, it would repress and stop the mRNA from also binding and transcribing the gene. It would be known as a repressor protein. So it is turning off the gene. That's a form of negative control. Other genes that are normally off, because the repressor is always bound, and so essentially you always have this repressor there, but there's an opportunity to move it, and so they would be referred to as inducible. In both situations, the genes are under negative control because repressor proteins are either always present and get removed, or not yet there and then get put there to turn off the transcription. Positive control would be the opposite of that. So you have some genes that are normally off, but they can be turned on. So we're not talking about repressors here. We're talking about activators. We have repressors and we have activators. So these are inducible genes. Other ones are normally on because the activator is there all the time. And so again, if you removed the activator, then they would be not on anymore. So in both situations here, the genes are under positive control because there is something, this activator protein, depending on what gene it is, it'd be different activator proteins, but this activator, and this is going to be helping the gene transcribe, the opposite of a repressor. So molecules that affect regulatory proteins are called effector molecules. And so you have the regular protein, but then you have these effector molecules that can affect what this protein does. So inducers, as their name would suggest, they work with activators. So they will induce an activator to do its job as an activator and help that RNA polymerase land and do its job. So you can imagine maybe we have this activator protein, but it in its current form is not going to act as an activator. Give it the inducer and it will change this protein in some way to make it essentially a functional activator protein, which then allows RNA polymerase to land and allows transcription to happen. So an inducer turns on an activator. Inhibitors turn off an activator. So again, the activator protein could be doing its thing as it normally does and allowing RNA polymerase to transcribe the gene, but there could be this inhibitor that gets into it, which is the opposite of an inducer, that basically unbinds the activator protein and therefore RNA polymerase is not going to be able to transcribe the gene itself because the activator protein isn't there to help it. So inducers turn on activator proteins, inhibitors turn off activator proteins. And these would be in different situations normally where you'd have different activator proteins being affected by different inducers or different inhibitors. Effector molecules for repressors. So let's look at the other side of the story here. Again, it's pretty much the opposite. Something that turns off a repressor is called an inducer. So there are inducers for activators and there's inducers for repressors. If you induce a repressor, you're turning off a repressor. It's like a double negative, which means you get transcription. A co-repressor helps a repressor protein. And so when you have a repressor protein that isn't doing its job as a repressor, and again, this would be each its own situation. So for this particular repressor protein, it can't bind to the DNA until it has some help from a co-repressor, and that would stop the transcription from happening. So to summarize this, we've got our 4 different situations here. We have inducers, which can either work on activators or on repressors, and we have inhibitors / co-repressors that again can either act on activators or repressors. So if we have an inducer working on an activator protein, we get an increase in transcription. So more transcription. The activator is turned on by that inducer. And so one example of this is CAMP can activate the lac operon. We'll see that as an example later. Also an inducer, so that an inducer turning on an activator gets transcription to increase. But an inducer working with a repressor, it actually turns off the repressor, which means you get an increase in transcription as well. So as the name inducer would suggest, you get more transcription. It can either get that more transcription by turning on an activator or turning off a repressor. A inhibitor with a repressor, you've got this inhibitor that is specifically a co-repressor working with a repressor that is going to turn the repressor on, allow the repressor to do its repressing, and therefore you would get a decrease in transcription. An inhibitor can also turn off an activator, and so you'd also get a decrease in transcription. If the activator was doing its job, then this inhibitor comes along and messes up its ability to do its job. You're going to see a decrease in that transcription.